Thank you so much, Dr. Subramaniam and Dr. Ahmed. So our first question, and I'll just say to the audience, make sure if you'd like to ask a question, you can go ahead and put it into the chat pod in the bottom right, and we will hopefully get to your question today. So our first question is for Dr. Subramaniam. How can industry help to make the review time most efficient for bioanalysis?
Hi, can you all hear me? Okay.
Yep, we can hear you.
Okay, so this question has been asked, and I put this right on top for obviously selfish reasons. This is a good question. So one of the things that first things first, I think you need to include all the validation and bioanalytical reports in the right areas. And also, as I mentioned during the talk, in the validation and the analytical reports, you need to mention any unexpected issues, and not only that, you need to address the impact of those issues. In addition, if the submission involves multiple assays or multiple iterations of the same assay, then it's good to include a summary description of the major changes to the assay chronologically, like why it was happening, how whether it was done to improve the performance or resolve a problem, what was the outcome, which studies used these assays, summary of the performance, and whether these assays were cross-validated and outcome. So a summary would be good to have, and this could go in module 2.7. And so these kind of issues would be very helpful for the reviewer, because typically, as I mentioned, when we review a new molecular entity submission, it's not just one assay. There are many assays. So it's in addition to the things that the clinical pharmacology reviewer does, he has to also he or she also has to look at bioanalytical. So this would be very important as far as trying to figure out what the problem is, what are the areas that one should focus on. So just remember, make sure you have the validation and bioanalytical reports in the right places, and then make sure the report contains all the critical information, and then also make sure that you have a concise summary of, the various iterations of the methods and multiple assays, the description, why it was changed, what was the problem, the outcome, what studies they were used in, and that's the performance. Thank you.
Okay, great. Thank you so much. And we have another question for you. How often should the method accuracy, how often should that be monitored? And where would that go in the validation report or the sample analysis report?
That's a good question. I think it goes back to one of the examples that I provided in the presentation. So there's no hard and fast criteria for monitoring the accuracy of the method, but one needs to keep in mind that just because you validated a method doesn't mean that everything is going to be fine during analysis, because things can happen, unexpected things can happen. So one needs to be cognitive of the fact and make sure that whether you're seeing anything unusual, whether there are any differences in concentration that you have observed now versus you observed earlier in other studies. And based on the totality of the data, one needs to make a decision whether the assay is performing as it's supposed to perform, it's doing what it's supposed to do, and take it from there. As far as reporting in the validation analytical report, I think you would report it. There's nothing, not a particular section, but usually one has, usually we see a section in the validation report or the analytical report that talks about unexpected issue or things that there are protocol deviations and so on and so forth. So you would actually put those kind of issues over there, or you can put it in any heading, as long as it's clearly identified in the report and the reviewer is able to look at it closely. Hope I answered the question.
Okay, great. We have another question for you about cross-validation. If the method if the method is revalidated for a new clinical study at a different CRO, does that require them to conduct a cross-validation?
As I mentioned during the presentation, cross-validation depends upon a number of things. So if you're cross-validating the assay, let's say the same, these assays are used in the same study or they are used across different studies, and if you're going to cross-compare these studies, or if these studies are going to be important for making decisions, as I mentioned, for dosing decisions or for labeling purposes, then I think it's important to cross-validate. So you have to take all these issues into consideration when you are making a decision to cross-validate or not cross-validate. And it's pretty much clear in the guidance as to what are the circumstances you need to cross-validate.
Great, thank you so much for those answers. And now we're going to go ahead and ask a couple questions to Dr. Hamed. So our first question. Hello. Hi, Dr. Hamed. Thanks for being here to answer questions and for a great presentation. So our first question for you, what are the requirements for biosimilars bioanalysis method?
Thank you so much for that question. Well, basically, as I highlighted throughout my talk, the requirements are basically what's highlighted in the bioanalytical method validation guidance. But in addition to that, we have some situations that are specific to biosimilars, given that we're comparing 2 or maybe 3 products in the PK similarity study. Some of the aspects we need to think about is what are the QC samples and calibration standards that are going to be used during the validation. And once these have been determined, these need to be used consistently throughout the PK studies that are conducted. And the bioanalytical similarity between these products needs to be established. That's one important thing. Also, one other thing is stability. Usually the sponsors, as we've seen in one of the examples that I've stated, they switch stability testing from one product to another, and this is not acceptable given that the 3 products, at the time of review, we think they are 3 different products. The stability needs to be established for each single product and cannot be extrapolated from one to another until a demonstration of biosimilarity is established. And also, I refer the more details are provided in the reference that are provided on slide 30.
Great, thank you for answering that question from our audience. So our next question for you is, why are there bioanalytical issues? Why are they more frequent than PK study issues with bio?
That's a very good question. One of the things about biosimilars from a regulatory perspective is that there is a lot of prescribed meetings. And the clinical development program is discussed in details in the design of the PK study, and the submission of the data is pretty much standardized in terms of like CDER and all of this regulatory requirements. So by the time we get to the BLA submission stage, the information is pretty much agreed with the FDA through communications. The design is discussed with the FDA and agreed. So there is very low potential for issues. As for bioanalytical, the information is often, as I showed in one of the pie charts that are shown in the presentation, often validation information is missing that we have to request during the review cycle. And as Sriram highlighted in his talk that was previous to mine, we have to issue these information requests during the review cycle. And there's a lot of questions that we find during the review cycle that are typically not found in the PK study because there was a discussion that happened between the FDA and the sponsors.
Great, thank you, Dr. Ahmed. Our next question for you is, what is the utility of PK data from the comparative clinical
Yes, so basically biosimilarity is established based on the PK similarity study. And then after that, there is a comparative clinical study that is needed for some products where a PD marker does not exist. And in that study, the objective of that study is to show the equivalence between the proposed biosimilar and the reference product. And in these cases, PK is collected for exploratory purposes. It's not for a demonstration of biosimilarity because that was already conducted in the PK similarity study. But the information that comes from the clinical study is basically used for exploratory purposes, like the effective immunogenicity on the PK of the product, some other quality issues that might affect the PK of the product. So we use the PK information from the comparative clinical study to make these assessments for exploratory purposes only.
Great, thank you. We have one more for you, and then we'll switch back to our other presenter. So what are anchor points, and how do they differ from the upper and lower calibration limits?
So usually the anchor points are they're treated like QC samples, but they're below the limit of quantitation on one side, and they're higher than the upper limit of quantitation on the other side. And basically, we use them to improve the fit, the 4-parameter or 5-parameter logistic regression for the calibration standards.
Okay, great. Thank you. All right, our next question is for going back to Dr. Subramaniam. Could you comment on incurred sample reanalysis? Are reviewers seeing the ISR information desired?
Yes, I think for the most part, we do get ISR information. So this is pretty much common right now, at least most of the studies that are being done recently. So this is more or less a fixture at the moment. And as far as incurred sample analysis, the expectation is that for any pivotal PK study, I think the incurred sample analysis is needs to be done. So yeah. Going back to the question about anchor points, I think Dr. Hamid mentioned about anchor points. And just wanted to stress that, so as Dr. Hamid mentioned, anchor points are basically used for improving the fit of the curve, of the calibration curve. That shouldn't be confused with getting the run into accept. So these are two different issues. So I think that we have seen people getting confused and have used anchor points for reasons other than that they intended use. So that's something one needs to bear in mind, that anchor points are just to improve fit irrespective of whether the run passes.
Okay, great. We have another one for you. Would it be acceptable if the blood stability test is repeated more than 2 times? How can we deal with repeated results?
Let me answer this in a more general perspective rather than limiting it to blood stability tests. So I think this can be common for any stability testing that we do. As I mentioned in the presentation, the key question that one needs to ask is when you're repeating a stability experiment, why are you repeating it? Are you repeating because you don't like the data? Or are you repeating it because you think that there's a problem? So thinking there's a problem is different from knowing that or finding out that there is a problem. So that is something that one needs to remember. So if you're thinking or if you want to retest, for some reason or the other, then I think you have to report all the information, because we don't know the problem. So you report everything, and then one can decide. and provide your decision and your rationale, and then we would also be able to figure out whether that's something the rationale is acceptable or is there some other issue that has not been looked at.
Great, thank you so much. All right, we're going to go back to Dr. Hamed. Could you please explain with any example that you see fit the parameter selectivity specificity for bioanalytical method validation?
Yes, the parameter specificity and selectivity are highlighted in the bioanalytical method validation guidance. And basically, selectivity is basically to assess if we can detect the analyte reliably in different serum samples, and if the matrix, basically to see the matrix effect. In terms of specificity, to see if the method can specifically measure the analyte that we're interested in, not any other interference like ADAs or concomitant medications or issues like that.
Great, thank you. We have another question for you. Could you please clarify several terms for the audience? Interchangeability, similarity, and equivalence from your specific perspective.
Well, basically, similarity is the objective of similarity is to prove that there are no clinically meaningful differences between the products, right? And then, as I mentioned in the slides, interchangeability builds on similarity, where basically the goal of the exercise is to establish that there are no additional risk or with interchange in the products, going from product A to product B and then going back to product A once interchangeability is established. So basically, in essence, if we exchange the products in single patient, we're not expecting to have any increased risk in terms of a loss of efficacy or increased safety profile. In terms of equivalence, basically, to me, equivalence is a statistical term that says that the product is not any worse or any better than the reference product.
Great, thank you again for answering so many questions. There's a lot of interest. So again, for you, Dr. Hamed, you mentioned that the PK is an endpoint for establishing interchangeability. What are the key differences in designing a PK study for establishing biosimilarity versus interchangeability?
Sure. So basically, a PK similarity study is specifically designed to detect any differences between the two products, the proposed biosimilar and the reference product. That could be like a single dose study in a patient population or healthy volunteers. And as I mentioned in the slides, the main objective of the PK similarity study is to detect the differences between the two products if they do exist, indeed. Interchangeability builds on the similarity, establishing similarity. The next step is actually to give multiple doses of the product. So as highlighted in the guidance for interchangeability product, it's a switchability study. So essentially we have two arms, one in which the reference product is being administered at multiple treatment cycles, and then the biosimilar product is being interchanged. There is a switching arm. And the idea is to see that there is no difference between the performance of the two products. And usually this is conducted in a patient population for which which the product is being approved. It is a multiple dose study, and then there is a thorough assessment of PK.
Great, thank you so much. And thank you for your presentation, Dr. Ahmed, and thank you for your presentation, Dr. Subramaniam, and taking